Transforming chlorinated waste into valuable synthetic organics. Unearthing the hidden potential of chlorinated waste. In a remarkable feat of scientific ingenuity, researchers have found a way to transform chlorinated waste into valuable synthetic organic compounds, effectively upcycling this often overlooked environmental burden. The ubiquity of chlorinated materials, from everyday plastics to industrial solvents, has long posed a challenge for disposal and recycling. Landfill, incineration, and even dechlorination methods have proven inadequate, often generating more hazardous byproducts. But now, a pioneering study published in Nature Chemistry has uncovered a game-changing solution. Inspired by advancements in catalytic chemistry, the research team led by Dyson and colleagues has developed a versatile PDCU catalyst system that can mineralize a diverse array of chlorinated waste, from PVC pipes to electrical wiring insulation. This process not only liberates the chlorine atoms but also generates a mixture of carbon monoxide and carbon dioxide, gases that are then seamlessly upcycled into valuable organic compounds. Through a series of elegant transformations, the team demonstrated the synthesis of over 30 aryl chloride products, including the active pharmaceutical ingredient vismodegib, a crucial medication for the treatment of basal cell carcinoma. Remarkably, this approach ensures that every carbon atom in the waste stream is meticulously repurposed, with no valuable resources going to waste. The sustainability and efficiency of this method shine through in a detailed life cycle analysis. When compared to traditional chlorination approaches, the researchers' process significantly reduces the carbon footprint, with up to 20-fold lower greenhouse gas emissions. This is achieved despite the fact that the method generates CO2 as a byproduct, underscoring the remarkable ingenuity of the overall approach. The old cliché of, one person's trash is another person's treasure, in this case rings true, the authors note, highlighting the transformative potential of this work. By harnessing the inherent chemical potential of chlorinated waste, the team has paved the way for a true circular economy in the chemical industry, where waste is no longer a burden but a valuable resource. As the world grapples with the growing challenge of plastic pollution and hazardous waste management, this study offers a glimmer of hope. It demonstrates that with creative thinking and scientific prowess, the seemingly intractable can be turned into the extraordinary, a testament to the power of innovation to reshape our relationship with the environment. Source: www.nature.com. Unlocking the power of catalysis, strategies for effective assembly. Unlocking the secrets of catalysis, a transformative breakthrough. In the ever-evolving world of scientific discovery, the quest to understand and enhance catalytic processes has long been a driving force. Now, a team of researchers has uncovered a remarkable insight that could pave the way for a new era in hydrogen production. The story begins with the intriguing puzzle of how metal-containing catalysts facilitate the conversion of water into hydrogen gas. For decades, scientists have grappled with this challenge, drawing inspiration from nature's own remarkable catalysts, enzymes. The breakthrough came when researchers delved into the molecular structures of these enzymes, revealing the presence of intricate metal centers that play a crucial role in the reaction. Inspired by these natural systems, scientists have sought to create artificial catalysts that can harness a wider range of metal ions, working alone or in tandem, to drive the hydrogen production process. However, the path to perfecting these catalysts has been fraught with challenges, until now. Enter the groundbreaking work of Miller and his colleagues, as reported in Nature Chemistry. Their research unveils a remarkable discovery: the self-assembly of multimetallic catalysts in water can dramatically enhance the efficiency of hydrogen production. By leveraging the inherent tendency of these catalysts to aggregate, the researchers have found a way to bring the active sites into close proximity, facilitating a remarkable synergy that boosts the catalytic performance. The key lies in the team's innovative approach to catalyst design. They started with a well-studied iridium-based catalyst, known for its ability to convert protons into hydrogen gas when exposed to light and an electrode. However, the efficiency of this process was limited by the need for two iridium sites to be in close proximity. The researchers then made a series of advancements. First, they created a dimeric iridium catalyst, where two catalyst units were tethered together. This proved to be a step in the right direction, but the true breakthrough came when they discovered that these dimeric catalysts actually self-assemble in water, forming small aggregates that serve as the active catalyst. The real game changer, however, was the team's subsequent finding that even simpler, monometallic iridium catalysts with hydrophobic tails can self-assemble in a similar manner, producing aggregates that outperform the bimetallic variants. This unexpected observation challenges the conventional wisdom that catalyst aggregation is inherently detrimental to performance. The implications of this discovery are far-reaching. By harnessing the power of self-assembly, the researchers have not only improved the catalytic efficiency but also managed to break free from the typical trade-off between rate and overpotential that plagues many catalytic systems. This feat is a testament to the team's innovative thinking and their ability to challenge established notions. As the scientific community grapples with the pressing need for sustainable energy solutions, this breakthrough in catalyst design could hold the key to unlocking the full potential of hydrogen production. The ability to create self-assembling, multi-metallic catalysts that can operate under mild conditions and harness the power of light-driven processes is a significant step forward. The road ahead is not without its challenges, as the researchers acknowledge. Questions remain about the underlying mechanisms that govern the solubility and stability of these aggregated catalysts, as well as the potential for extending this approach to other catalytic systems. Nevertheless, the foundations laid by this groundbreaking work have set the stage for a new era of exploration and innovation in the field of catalysis. As the scientific community eagerly awaits the next chapter in this captivating story, one thing is clear: the pieces are falling into place, and the path to improving catalysis has just become a little brighter. Source: www.nature.com. Public health priorities following elections in the DRC. Here is a 500-word compelling and engaging story summarizing the provided text, written in a human-like style with a high degree of burstiness, a glimmer of hope amid the darkness, the DRC's post-election responsibilities for public health. As the Democratic Republic of the Congo, DRC, emerges from a tumultuous election, the country faces a daunting reality, a severe humanitarian crisis that has been compounded by years of armed conflict, political instability, and economic hardship. The tensions in the DRC have reached a boiling point, with the Congolese army and its allied militias battling the March 23 movement group in the volatile regions of North and South Kivu. The consequences of this escalating conflict are devastating. The United Nations has reported over 3,000 human rights violations since October 2023, including the arbitrary murder of civilians, abductions, and the forcible recruitment of children into armed groups. Coupled with a crippling food crisis that has left 25.8 million Congolese facing food insecurity, and the recent outbreak of a lethal clade Mpox virus, the DRC is facing a perfect storm of humanitarian catastrophes. Yet, amid this darkest of hours, a glimmer of hope emerges. The re-election of President Felix Tshisekedi's administration presents an opportunity for the DRC to end the relentless cycle of suffering and chart a new course towards stability and prosperity. And as a leading science journalist, I believe that a public health-centric approach can provide the roadmap for this crucial transformation. First and foremost, the international community must step up and provide the necessary funding and resources to address the immediate crisis. With the UN's humanitarian response plan only 37% funded, the DRC's most vulnerable populations are being deprived of essential aid. Increased media attention and social media campaigns can help drive donations to relief organizations like UNICEF, enabling the rapid deployment of food, medical supplies, and personnel to the hardest-hit regions. Simultaneously, the DRC government must work tirelessly to secure aid and protection for the millions of internally displaced persons and refugees who have fled the country. By forging secure supply routes and establishing shelters connected to central hubs, the administration can ensure that these displaced populations have access to the basic necessities and trauma-informed care they so desperately need. Looking beyond the short term, the DRC must prioritize long-term solutions for stability and peace. This will require a regional diplomatic offensive, where the DRC allies with its neighbors to reinvigorate the stalled Nairobi and Luanda processes and initiate direct negotiations with Rwanda and rebel groups. Crucially, this coalition must also work to ensure the responsible management of the country's valuable mineral resources, safeguarding against foreign exploitation and channeling the revenues towards local redevelopment. Finally, a stabilized DRC must turn its attention to rebuilding and strengthening its healthcare system. By emulating Rwanda's successful community-based health insurance model, the DRC can expand access to comprehensive primary care and establish robust disease surveillance systems to tackle the ongoing challenges of cholera, mpox, malaria, and yellow fever. The road ahead is long and arduous, but the re-election of President Tshisekedi offers a glimmer of hope for the DRC. With a steadfast commitment to public health, a renewed regional diplomatic effort, and the unwavering support of the international community, this embattled nation can finally break free from the shackles of conflict and emerge as a beacon of stability and prosperity in Central Africa. Source: www.nature.com. Older nasal tissue linked to increased risk of severe COVID-19, unraveling the age-old mystery, how nasal epithelium shapes COVID-19 severity. As the COVID-19 pandemic continues to ravage the globe, researchers have remained steadfast in uncovering the intricate mechanisms that underlie disease severity. Amidst the chaos, a team of scientists has uncovered a remarkable insight: the age of an individual's nasal epithelium plays a crucial role in determining their susceptibility to severe COVID-19 outcomes. The study, published in the prestigious journal Nature Immunology, sheds light on the remarkable differences in the composition and function of nasal epithelial cells, NECs, across various age groups. Woodall and colleagues meticulously cultured NECs from children, adults, and older adults, and discovered that the cellular landscape of these vital guardians of the upper airway varies significantly with age. In pediatric NECs, the researchers identified an expansion of a specialized cell type known as goblet inflammatory cells. These cells possess a remarkable ability to mount a robust antiviral response, characterized by the robust induction of interferon-stimulated genes, ISGs, and the production of type 1 and II interferons, the body's most potent antiviral weaponry. This intrinsic immune response appears to be a hallmark of the young, as it effectively limits viral replication and spread, preventing the virus from reaching the lower airways and causing the dreaded acute respiratory distress syndrome, ARDS. In stark contrast, NECs from older adults tell a different story. These cells exhibit a heightened pro-repair and pro-fibrotic landscape, with the emergence of a unique, bacilloid-like, cell population expressing markers associated with wound healing and tissue scarring. Intriguingly, these aged NECs demonstrate an increased capacity for viral spread, with augmented production of infectious viral particles and a compromised epithelial barrier. The authors propose that this age-dependent cellular makeup of the nasal epithelium plays a pivotal role in shaping the clinical trajectory of COVID-19. While the robust antiviral response in children may limit viral dissemination, the pro-repair phenotype of older adult NECs may inadvertently facilitate viral growth and spread, contributing to the severe outcomes observed in this vulnerable population. Importantly, the findings from this meticulous in vitro study are corroborated by analyses of publicly available single-cell RNA sequencing data from the upper and lower airways of COVID-19 patients, underscoring the translational relevance of these discoveries. As the COVID-19 pandemic continues to evolve, with the emergence of increasingly formidable viral variants, the insights unveiled by Woodall and colleagues take on even greater significance. Understanding the age-dependent dynamics of the nasal epithelium and its impact on antiviral immunity may pave the way for the development of targeted interventions, tailored to the unique needs of individuals across the lifespan. In the ever-changing landscape of the COVID-19 crisis, this study stands as a testament to the power of scientific inquiry, revealing the intricate interplay between age, cellular composition, and the body's ability to mount an effective defense against this relentless foe. As we continue to navigate these uncharted waters, the lessons learned from this study will undoubtedly contribute to our collective understanding and, ultimately, our ability to safeguard the health and well-being of all. Source: www.nature.com. Unlocking the role of transcription factors and SWI-SNF complexes in chromatin priming during T-cell development. Unveiling the epigenetic symphony, how T-cells orchestrate their destiny. In the ever-evolving world of immunology, a fascinating discovery has emerged from the research labs of scientists at the University of California, San Diego and the Salk Institute for Biological Studies. Their findings, published in the prestigious journal Nature Immunology, shed light on the intricate dance of transcription factors and chromatin remodeling complexes that orchestrates the development of specialized T-cells. The journey of T-cell maturation is a captivating tale, where progenitor cells in the thymus navigate a complex landscape, ultimately differentiating into distinct effector subpopulations. At the heart of this process lies the concept of chromatin priming, a pre-established chromatin state that prepares the stage for future transcriptional responses. Gamble et al. Have uncovered the molecular mechanisms that govern this pivotal step, revealing a harmonious interplay between key players. The study begins with a surprising discovery. Chromatin priming at loci associated with effector T-cell genes occurs remarkably early in T-cell development, even before the expression of the T-cell receptor, TCR. This finding challenges the conventional understanding that TCR signaling is the primary driver of chromatin remodeling in T-cells. The researchers delve deeper, identifying the triumvirate of transcription factors, PU.1, RUNX1, and BCL11B, as the maestros orchestrating this chromatin priming symphony. These factors, expressed at distinct stages of T-cell development, work in coordination with the SWI-SNF chromatin remodeling complex to establish and maintain the poised enhancer landscape. In the early stages of T-cell development, PU.1 and RUNX1 direct the SWI-SNF complex to key effector T-cell loci, initiating chromatin accessibility and the deposition in the H3K4Me1 histone mark, a hallmark of poised enhancers. As T-cells progress towards lineage commitment, PU.1 is silenced, and the baton is passed to BCL11B, which teams up with RUNX1 to sustain the primed chromatin state. Intriguingly, the authors unveil a collaborative competition model, where RUNX1 forms distinct complexes with the SWI-SNF complex and BCL11B, alternately occupying the same genomic regions to reinforce chromatin accessibility. This dynamic interplay ensures the robust maintenance of the primed enhancer landscape, even in the absence of PU.1. But the story doesn't end there. The researchers also uncover a surprising duality in BCL11B's role. While it is essential for maintaining chromatin accessibility at a subset of effector T-cell loci, it also acts as a repressor, preventing excessive chromatin remodeling and SWI-SNF binding at regulatory regions associated with alternative cell fates, such as natural killer cells and innate lymphoid cells. This elegant study not only elucidates the intricate choreography of transcription factors and chromatin remodelers in shaping the epigenetic landscape of developing T-cells but also paves the way for a deeper understanding of how these fundamental processes contribute to the diverse functions of mature T-cells. As we continue to explore the frontiers of immunology, discoveries like these hold the promise of unlocking new therapeutic avenues and advancing our ability to harness the power of the immune system. Source: www.nature.com. Improving Fairness in Medical AI with Unlabeled Data, Unlocking the Power of Unlabeled Data, AI's Path to Equitable Healthcare. In an ever-evolving world of medical advancements, the promise of artificial intelligence, AI, has captivated the healthcare industry. However, as these AI models are trained on limited, specific datasets, a pressing concern has emerged: their ability to generalize and perform equitably across diverse patient populations. Rajiv Mova, Pang-Wei Ko, and Emma Pearson, three leading experts in the field of medical AI, have shed light on a groundbreaking approach that harnesses the power of unlabeled data to enhance the fairness and generalization of these clinical algorithms. The key lies in the recognition that while labeled datasets used for training may be small and narrowly focused, the wealth of unlabeled data available can hold the key to unlocking more robust and equitable features. Vaidya et al. And Keitena et al., in their respective studies published in Nature Medicine, have demonstrated the transformative potential of this approach. Vaidya et al. Found that by replacing a non-medical image encoder with one trained on a vast, diverse dataset of pathology images, their model for predicting cancer subtypes was able to significantly reduce disparities in accuracy across racial groups. The pathology-specific encoder had learned a richer feature set, rendering it more resilient to distribution shifts. Keitena et al. Took a different approach, leveraging unlabeled data to generate synthetic images that were then used to augment their original training dataset. This not only improved overall performance but also mitigated accuracy disparities across sex and race groups, a testament to the power of diverse data representation. These studies underscore a critical insight: we can no longer rely solely on small, labeled datasets to train the AI models that will shape the future of healthcare. By tapping into the wealth of unlabeled data, we can empower these models to learn more generalizable and equitable features, paving the way for a future where AI-driven medical solutions truly serve the needs of all patients, regardless of their demographic or geographic background. The implications are far-reaching, as these approaches hold the potential to address the long-standing challenge of bias and lack of generalization in clinical algorithms. As Mother, Co., and Pearson emphasize, the continued collection and curation of large, diverse datasets, annotated with relevant clinical and demographic variables, will be crucial in driving this transformation. Ultimately, the journey towards equitable and robust medical AI is one that demands a holistic approach, embracing the synergies between labeled and unlabeled data, as well as the collective efforts of clinicians, computer scientists, and regulators. By harnessing the power of unlabeled data, we stand poised to unlock a new era of healthcare solutions that truly leave no patient behind. Source: www.nature.com. Unveiling the transformation of Australia's Great Barrier Reef through coral bleaching, unraveling the shifting sands of the Great Barrier Reef. In a startling revelation, marine biologists have sounded the alarm on the profound transformation taking place within Australia's iconic Great Barrier Reef. The world's largest coral reef system, a natural wonder and UNESCO World Heritage Site, is undergoing a dramatic shift, driven by the relentless onslaught of climate change. The culprit? Repeated coral bleaching events, a consequence of rising ocean temperatures, have been decimating the reef's vibrant ecosystems. A recent report by the Great Barrier Reef Marine Park Authority paints a grim picture. 3/4 of the entire reef is showing signs of bleaching, with nearly 40% experiencing high or extreme levels. It's not a question of reefs dying or reefs disappearing, it's reef ecosystems transforming into a new configuration, warns marine biologist Terry Hughes from James Cook University. As the corals, the foundation of these underwater habitats, succumb to the stress, the ripple effects are felt throughout the entire ecosystem. The iconic fish, crustaceans, and other marine life that depend on the coral's intricate structures are facing an uncertain future. The situation is further exacerbated by the frequency of these bleaching events. In the last 6 years alone, the Great Barrier Reef has experienced 3 mass bleaching episodes, leaving little time for the corals to recover. We've settled into bleaching every other year, in 2020, 2022, and now 2024, and that's simply not enough time for a proper recovery, laments Hughes. The transformation is not limited to the Great Barrier Reef, it is a global phenomenon. The United States National Oceanic and Atmospheric Administration has declared the 4th global coral bleaching event on record, the 2nd in the past decade. From the southern hemisphere to the northern, the warming waters have taken a devastating toll on these delicate marine ecosystems. There have been very high temperatures driven by climate change all across the world, and there has been coral bleaching in many other countries, says environmental scientist Roger Beeden, chief scientist for the Great Barrier Reef Marine Park Authority. As the corals succumb to the relentless heat, the ecosystem's dynamics shift. Fast-growing, branching, and table-shaped corals, which are often the first to recover, are also the most vulnerable to bleaching. Their resilience, ironically, undermines the reef's ability to withstand the next inevitable bleaching event. Seaweeds, on the other hand, flourish in the degraded coral habitats. The future of the Great Barrier Reef remains uncertain, but one thing is clear: the solution lies in the hands of humanity. Reduce greenhouse gas emissions. Full stop, urges Hughes, the marine biologist. The fate of this natural wonder, and the countless species it supports, rests on our ability to address the root cause of the crisis, climate change. Source: www.nature.com.